that
Sorry.
That's okay.
I just realized I forgot to record.
That like the work can potentially be dangerous for the people who are doing the work, right? As a lot of research can be if you're using radiation or if you're using, right? So, but the work can potentially be dangerous. And that also, you're playing with the building blocks of life, and that raises some concerns and some questions about what your motivations are and what you're trying to do and what effects that might have, and so on. So there's been this kind of, this conversation has started a while ago. And then in the late 1990s, in the late 1980s, I'm getting my dates wrong. The Human Genome Project got underway. So this was this, obviously we all know this push to sequence the human genome. There's an interesting backstory to it that I don't know if you're all aware of, but has anyone heard of Craig Venter? He's a fairly infamous scientist by now. He's pretty big in the world of synthetic biology. He is his institute, the J. Craig Venter Institute in San Diego, is where they created the first completely synthetic organism. So they basically synthesized a genome of a mycoplasma from scratch, and then booted up, like they took the genome out of another species of mycoplasma cell and they put their synthesized genome in, and they booted it up, and they showed that it took on the characteristics and the properties of the species that they had synthesized. This was, I'm trying to think when this was, maybe 2006 or 7, somewhere around then, maybe 2008. But anyway, he also was deeply involved in the Human Genome Project. And actually, the reason that the Human Genome Project took on the form that it did is really because of Craig Venter. So he had already made money. He was running a company called Celera, and he decided he was going to sequence. And that got a lot of people kind of worked up, and in fact got the British and American governments so worked up that they decided basically to race Craig Venter to sequence the human genome. So the reason the Human Genome Project took the form that it did was because governments basically said, no, this information has to be public domain. We don't want a private company synthesizing or sequencing the human genome and then locking it up. And in the end, again, I can send you, there was something recently even about it. It was just very odd. So Bill Clinton was president at the time when the Human Genome Project was finished. And literally he held this press conference with the head of the NIH and Craig Venter side by side, who agreed that it had been a tie and that they were going to release the human genome sequence kind of publicly. And there was a recent piece, I think, in Nature saying what an odd moment that was, just to sort of have the sort of whole backstory and whatnot. Anyway, Craig, yeah.
So since these governments were concerned about it being private property, did this Craig Venter person have any intention, because to me it seems, obviously science is very collaborative and people would be eager to share things like this. Did he have any intentions of keeping it shut away like that, or?
Yeah, I mean, he basically had declared the intention that his company was going to sequence the human genome and file for patents on it so that it would be locked up, which is basically why I think the governments and the national governments got pretty worked up about it. So he, in a sense, single-handedly spurred the Human Genome Project in the form that it did, right? But one of the interesting things, so government, the British and American governments in particular, poured a lot of money into the Human Genome Project. And again, it's kind of an interesting story of how they sort of tried to race to get the genome sequenced. But they decided at the time that they were going to allocate 5% of the funding for the whole Human Genome Project to studying some of the ethical, legal, and social implications of sequencing the human genome, because this was what are we going to discover about humanity through this process? How is it going to change the practice of medicine? What's it going to, legally? I mean, it was clear Craig Venter had raised the issue of can you patent and own and commodify the human genome? that there were a lot of potential kind of questions and issues that might be brought to light by doing this kind of work. And so basically since then, so since the Human Genome Project, for a lot of new and emerging technologies, there's been this tendency by research funders in Europe and the US especially to allocate some portion of the funding, and 5% has become the sort of the default number, to allocate some percentage of their funding to studying some of the broader impacts of the research. So this holds in nanotechnology, and I think in a lot of the kind of brain type initiatives, a lot of the really big neuroscience initiatives. I think some funding has gone towards it gets shortened to ELSI, ethical, legal, social, has gone towards ELSI in geoengineering, so some of these new and emerging technology areas, there's been this kind of tendency or this habit then to carve off a little bit of the funding to research some of these broader questions. Now, fast forward to the sort of advent of synthetic biology, which really started taking shape around 2000 or so, is when, there were some serious calls for funding and things like that going out. And 2005 is when the NSF in the US made its first big investment in synthetic biology. So they set up a center called SynBurk, which was distributed across a number of universities, but a lot of the big players that you might expect. So I think it was Stanford, Berkeley, MIT, Harvard. I think there was a Texas college and maybe one or two others. And so again, when setting up this kind of big institute, they decided to allocate some money to doing this kind of work. Actually, one of the main people who was involved in setting up what that work looked like at SynBurk is at ASU as well. His name is Gaiman Bennett. He's an associate professor in Shippers, which is the School of Religious, Philosophical, and Historical Studies. So he was like an embedded theologian and anthropologist in that center when it was set up for several years. So if you're ever interested, you could probably bring him in to chat about that experience. But one of the things they did, so actually Gaiman is responsible for the naming human practices, which is what you see in iGEM. And is typically what this ELSI research has been renamed in synthetic biology. And the reason, there's a kind of, again, a story about why the sort of ELSI label had, at least for some group of us, sort of fallen out of favor a little bit. So with the Human Genome Project, it was ethical, legal, social implications. So what basically it was saying is this work is going ahead, and it's up to you guys to study like the impact, the fallout, the consequences, but not necessarily be involved in the shaping of the projects themselves. So the work that was done with the Human Genome Project was kept very separate. It was like historians and philosophers who were sort of, literally didn't interact with the teams that were actually sequencing the human genome. It was sort of separate work. I think the lawyers and the ethicists were maybe a little bit more involved, like directly involved in perhaps working with some of the scientific teams. But there was some sort of flack that they got, this kind of ELSI project has been criticized a little bit for like, it's just studying the impacts, just downstream. It's not, doesn't actually shape the science in any way. So the idea of human practices was like a way of reframing and escaping that label a little bit and suggesting a new kind of model where the work that, this kind of attention to, the relationship between a technology and the world it's designed for is really kind of part of the project itself, is kind of what motivated the term human practices. And so that's what you see in synthetic biology and in iGEM in particular. So, from pretty early on in iGEM, maybe 2 or 3 years into it, they introduced a sort of human practices component to the competition, which I think at the time was sort of optional. It was one of the things you could do to get a gold medal. And it has gradually become a more and more important part of the competition, such that now, actually, I haven't looked at the rubrics for this year, but it's mandatory. And so it's mandatory to get a silver medal. I'm not sure if it's mandatory to get a bronze medal. I don't think it is. But it's become a sort of more and more integral component of the competition, with an interesting backstory there. So, personally, I've been involved in it for a long time now. And I ran the human practices part of iGEM for a year. So you've got some in-house experience on hand if you if you want to at any point reach out. I'm actually not judging the competition this year. I think it's happening right now, like this week. But I have judged the competition probably 8 or 9 times. So I have a really good sense of what teams do, what good human practices work looks like, and just sort of how to avoid some of the major pitfalls that teams often fall into as well. Which is not, I think it's not about the teams per se. It's that, this work can be hard to do, and actually to have someone on your team who has a active interest and maybe some experience, and to have mentors around is super helpful.
Would you mind?
I realized I've been rambling. Was that?
No, you're good. I was wondering if you would mind sharing some of the interesting human practices, or maybe the fun backstory that came around.
Yeah, I mean, there's lots of, so early on, so there's one that's a microbiome story, if you want, one of the earliest kind of human practices examples. And I'm trying to think if it was maybe 2008 or 2009. And actually, so this was the Cambridge team in the UK, and they decided for their iGEM project, it was a sort of foundational advance. You can pick lots of different tracks to go into now. I don't think those tracks existed at the time. The competition has scaled so much since the early days. But the Cambridge team did something that turned out to be pretty foundational. Is they decided that they so many of the readouts that people use when they're doing assays to test for whatever in your cell cultures rely on fluorescence. And they wanted to move away from fluorescence and see if they could get cells to generate pigments instead. So they basically they synthesized, and they did it. They managed to create a rainbow set of pigments in their E. coli by reconstructing the metabolic pathways for different carotenoids, and I can't remember all the different pigment names, but it was pretty awesome. They did this kind of really interesting technical advance, but they also brought in 2 designers who are not your typical product designers, but came from a world called speculative design. And what they did was they led the team through a whole set of exercises to imagine the future of what this technology could do. And so they had different time points into the future, and sort of imagining what a world where you could have bacteria that give you colored pigment readouts looks like. And so, trying to think, they maybe had by 2030, you can have bio art tattoos that reflect your mood and things right? They're just sort of entering this speculative fiction. But this was really, at the time, right? This isn't typical for a science project, right? This is not, certainly not what you do at high school, and, isn't typical for other sort of robotics type competitions, which is what iGEM is sort of modeled off of. So they invited in these designers who then also came up with their own product. And I remember they were walking around the Jamboree in trench coats, carrying a briefcase, a metal briefcase. And they, what they said was that they had, I forget, it was maybe 2015 or 2020. So we've probably passed the time frame now. And they're okay, so, this technology develops. And what you can do is you can develop tailored probiotic drinks for individuals based on their genetic susceptibilities to different medical conditions. So if you are particularly susceptible to getting colds, and there's a history of colon cancer in your family, and you maybe have pre-diabetes markers and things like that, you could get this kind of customized yogurt drink where the bacteria in the drink were sort of programmed to detect very early markers of these different conditions. And they would respond by changing color. So in this suitcase, they basically had a box of colored poop, right? So that, like, you know, if your poop was blue, you knew you were like in the early stages of a cold or whatever, right? Or you had to get yourself to a doctor to, you know, right? But like this was pretty, I don't know. I mean, it sort of took the competition by storm because they had sort of done this kind of work. And I think they won, like the Cambridge team won the sort of grand prize that year for doing that. So that's like one, you know, that's kind of one example that set a bar for the kinds of things that that get incorporated. Well, Christina, this is a very good question. It turns out that at least in the UK, because this is where the team was based, one of the tabloid magazines, I'm trying to think what an equivalent of it's like one of the really big newspapers reported that this was an actual product. Like didn't, you know, but no, seriously now there are labs that are working to develop this. Like absolutely.
I'm going to tell you, this sounds painfully similar to the colon cancer idea.
I was like, you know, your microbiome idea has a, you know, has a lively precedent within competition. And I would definitely advise you to look at what I think it might have been Cambridge 2009. I can't quite remember what they did and some of the follow-ups. The problem with the iGEM website is that it's super hard to like, there's no easy catalog to navigate. So I can't, you know, there isn't a nice lineage where you could look at other teams who followed up on that. But absolutely, there is this push for like probiotic drinks that serve as real-time biosensors. But of course they wouldn't, you know, and the team wouldn't have thought of this necessarily, right? This was a sort of process of, yeah, you would. You totally would. Look, they're super proud that everything is open source. But yeah, no, it's.
I send people the Wikipedia page to explain what it is instead of the link to the actual website, because like just finding what it is on the website is like harder than the Wikipedia.
Mm-hmm. Absolutely. No, it is a mess. And actually I have a colleague who's been like trying for years to just like get like the entire sort of data dump from them so she can start to mine and look for, you know, start to like pull together, you know, themes and projects and stuff. And this is not to, this is being recorded, right? I was gonna say the head of the iGEM Foundation has very strong views about many, many things. And so you can complain about the website as much as you like, but he's not.
That's okay. We average like 4 views.
For them.
Yeah.
And we'd win.
Right. So
I'm saying.
Yeah, so there's a, oh, nice. Yeah. There you go. So yeah, the project was called eChromai, I think, in the end. You know, they had beautiful, because they had designers on board, they like designed their project beautifully. And it was just like this sort of, and actually the designer who was involved, Alexandra Daisy Ginsburg, is now like a well-known designer in this space who does a lot of work with emerging technologies and in particular synthetic biology. Sort of designing these, or thinking about not creating products, but sort of designing imaginative futures where you sort of are forced to grapple with the technology in interesting ways. So that's one example, but there's a whole bunch of, I mean, there's teams that have done sort of philosophical explorations. I remember one team fairly early on was very interested. They were trying to create a new chassis, you know, because there's like certain model organisms that get used a lot, and they were trying to create a new chassis. They got super interested in what the idea of a chassis was, and they did like a philosophical exploration of what that was. There was a team in Mexico that really struggled. The different like import-export laws around DNA are different in different parts of the world. And I don't think they were actually able to get the DNA from iGEM to start the competition. And so, or at least maybe it was held up for a couple of months. So they ended up doing like a sort of deep analysis of the regulatory frameworks in their country and working with legislators to try and come up with a proposal for, you know, how to address this issue. There was a team in China one year. These are sort of early examples that wanted to test. There were a few scandals early on around being able to, around DNA synthesis companies. And so somebody, this might've been like 2006, 2007, some reporter basically ordered sequences, DNA sequences from smallpox from a DNA synthesis company and got them sent to his home address, and like did an expose being like, you can just order smallpox and get it sent to your house, what's going on? And that sort of forced a big sort of reckoning with DNA synthesis companies, where now they have, it's not mandatory, but they have like software for screening, you know, against known pathogens and things like that. But there have been teams that have like stress tested that system in a sort of white hat hacker way. And I would say increasingly over the last few years, there's been a trend towards, and this is now rewarded in the prizes too, towards what's being called like integrated human practices, which is, I think for those of you who are certainly biomedical engineers, is probably something that's not going to be incredibly foreign, but isn't very common in the life sciences, and I don't know about in some of the other engineering degrees, but where you are doing, where your project is crafted with a particular set of stakeholders in mind that you engage with over the course of your project, to help you think through the project, to help you refine your ideas, to help you, you know, give feedback and so on. And that there's a sort of process by which you can kind of document how your engagement with stakeholders has shaped the project that you've done. And I would say that's at the moment sort of the dominant strand of human practices. I'm trying to think of teams that have done a really
last meeting, I think at the FAQ session, one of the graduate students told us about, what is it? Was it Canada? They
the
were
Canadian
dealing
teams
with,
are pretty kick-ass in this, yeah.
yeah, they were doing something with like these seeds. They would turn green too early or something, and they wouldn't actually grow to harvest. So they were actually working with all these farmers.
Yeah, they connected the entire chain of stakeholders, and I'm trying. It might have been Lethbridge. The Alberta teams, for whatever reason, I don't know if it's the same this year, but for like 6 or 7 years, there's been an Alberta foundation that has sponsored iGEM teams, and they've like brought them all together for boot camps for weekends. They flew me out one year to work with the teams on human practices. Like they really have invested a ton in sort of helping the teams thrive and have the mentorship that they need. So the Alberta teams, it's worth looking at them regardless. Like, you know, the website's so hard to trawl, but like Lethbridge, Edmonton, I don't know, there's a handful of Alberta teams that like always do well. I remember one group was working with like emergency responders to do something to do with disinfecting ambulances. I can't remember the details, but they might have even been a high school team. But you know, just like sort of really integrated work. I think the UC Davis team from sometime in the last 3 or 4 years, they all start to blend together a little bit. Did an interesting human practices project around olive oil, because apparently a lot of olive oil that you buy from the grocery store is rancid. And so they were trying to do like a olive oil rancidity test, I think, or trying to develop as their, you know, as their sort of more technical project. And they did a lot of work with stakeholders. Yeah, they did a lot of work with stakeholders like, you know, farmers and grocery stores and things like that, to sort of really try and diagnose what the problem was and think about ways in which they could sort of assist with it. So there's, you know, there are, I mean, there are some examples on the iGEM website of kind of good work. And then there's sort of depending on the types of projects that you end up choosing. But I would say finding some sort of interesting, clear connection to your project. And it doesn't have to be a real world. Like I was saying, you know, it's great if you've got a problem that's a local homegrown problem, because that gives you very quickly sort of something on the ground to investigate and people to talk to and so on. But you don't have to do that kind of work. I mean, you know, teams that are doing kind of much more foundational advances also do human practices work. But it, you know, you have to figure out, it's a combination of something that you're actually interested in, passionate about, and willing to put some time into. And then also kind of what ticks the boxes for the competition. The competition is very much a box ticking exercise if you do go down that route. although I've heard through the grapevine or actually talking with the head of iGEM last year, the plan is, I think, for next year, although I don't know if that's changing because of COVID, they are going to change it completely. I don't know what that means for iGEM. I think certainly talking with Randy, his thinking a year ago was to orient the competition around sustainable development goals. So actually, at least a couple of the projects I think would very clearly tie into sustainable development goals. And health is also clearly a sustainable development goal, right? So, yeah, so you sort of have to see what the competition rules are. And I don't know if they're going to change it in time, for next year. The plan was to move it to Paris and do sustainable development goals. So you might get a trip to Paris.
Maybe. Would you mind telling us kind of just like about, I guess, the path that you took into synthetic biology? Because it's a little different than, I don't know, not like that you're not a scientist, but the
Sure.
lab work kind of route.
The lab work, yeah. I mean, well, so I did train as a scientist, I did, and I did nothing but science. Maybe that's what I sort of was too extreme for me. So I did my undergrad majoring in neuroscience, and then my PhD was more of a biochemistry PhD, really looking at like signaling pathways, and so I was doing sort of a combination of molecular biology and biochemistry for my research, looking at white blood cell migration. But I sort of realized, and for those of you who are in the lab or planning to be in the lab or spend long periods of time in the lab, for me, I realized that like the questions about science and engineering that were super motivating to me were not the how am I going to design my experiment tomorrow questions. I did a ton of cell culture during my PhD, like a ton. And I was like, when I was able to freeze down and kill the rest of my cells, I was like, I felt such relief at the end of it. But I was always kind of interested in these bigger kind of questions about the politics of science and questions of access and questions of like who decides what gets funded and what counts as good science, and the sort of the rat race of like pressure to publish, and just all those kinds of questions were the things that I found myself thinking about, but not having any clue how to actually work on them. And I think this sort of encapsulates human practices too, is it's like there's this competition and you have to do this. And there are absolutely robust ways to do this, and there are also incredibly flimsy ways to do this. And you sort of muddle your way through. And so, but for me, I went and worked in science publishing after my PhD. I was like, oh, do I want to go do a postdoc? I don't know. I didn't necessarily want to like shut the door completely. But I got an internship at Nature, which is one of the really big science journals, and spent actually, so it was 6 months, but it turned into 2 years there. So I spent 2 years there rewriting or editing people's manuscripts. So I didn't get to make like editorial decisions, but I got to see the editorial process, which was super interesting and eye-opening for me. So like there is a big politics to science, especially if you're a big journal. So a journal like Nature actually doesn't even peer review 90% of the papers that get sent there. So the editors make the decision about which 10% they will actually send out for peer review, and the rest they just return based on editors saying, no, this isn't the kind of science we publish here, which is like, that's huge, right? And so I would get the packets, like, I would only see the papers that had been accepted, and I would get like the whole stack on my desk that would have all the correspondence relating to the manuscript. And you'd see conversations among the editors, like in the sidebars of like, yeah, well, if we don't send this paper out for review, we're never going to hear the end of it. And that was usually in relation to an alpha male who, was a big name in the field and things like that. So anyway, that whole experience got me really interested. Sci-Hub, is this so is Sci-Hub an explicitly open source? Actually don't know much about Sci-Hub.
David, do you want to explain, or? Oh, okay.
A pirating service.
Yeah.
I mean, there was something I think that came out this week in Nature about how most scientists use access pirated papers. Like, and maybe it's sci-hub. They were just like, yeah, I mean, this is what, you know, it's a really interesting and complex question. At the time that I was working at Nature, the PLOS journals were setting up and becoming a sort of, they were like an initiative to be, you know, where the paywall is not for the readers, but the authors pay to publish. And they were setting themselves up in strong opposition to the traditional journal publishing model. And, you know, it's a really interesting question. Like a journal like Nature decided to stick by its, you know, model. Yeah, I mean, as an author, you can send papers, you can, you know, I mean, there is a sort of currency. It's also common practice to email your friends at different institutions to see if they have access to a particular journal. So I'm very often doing that to my European colleagues because they often have this different set of subscriptions. So it's like, hey, could you find this for me? It depends. I mean, there's also this sort of growing kind of predatory journal landscape as well. So what the really big kind of old prestigious journals will tell you is that, like, actually there's a certain amount of reputation and quality and things like that they want to uphold. I mean, there was a staff of, I'm trying to think, there were 5 of us who worked full time to get every single paper ready for publication. And that wasn't doing the artwork. So there was an art team as well. And so like we basically, you know, they're offering their aim there was to say it shouldn't matter what your native tongue is. You know, the science should stand by itself, and that we basically had to rewrite all the papers. There's like a 150-page style guide that you sort of had to memorize to start the job, where you rewrite the papers to make it so that the language kind of falls out and you just read the science. So that there isn't really like you know, you don't get different authors have different flares when they write, that you try and strip all that out. And it's just very much a presentation of the science. You know, they argue that that's some sort of added value. Other people disagree, right? There's a lot of professors now who will refuse to publish anywhere other than open access. It's a complicated, it's a bit of a complicated mess. But it doesn't seem, you know, and there are universities now that are boycotting presses, right? Like I think Elsevier might be that the entire University of California system is no longer paying for any Elsevier journal as a way to try and push them. I guess the question is just, where do you get the funding from to publish the research? And it has to come from somewhere. I'm not saying that it's the readers who should pay. It's also not clear that can always be the authors who pay. Increasingly, they're turning to funders. So like basically if you apply for grant money and you get a grant, that the institution that's funded the research should fork out, but it's usually $3,000 or $4,000 a paper to publish it. So yeah, and anyway, so sorry, this is like a long, you know, winding history, but that experience at Nature got me really interested in like the politics of science. And so I thought, okay, I don't think I'm going to go back to the lab bench now. And so I retrained. I found a new, like, I was like, oh, I don't think I want bioethics. I'm not really into ethics. Not that I like, you know, it's like I like to think I'm a fairly ethical person, but sort of and I still hold, I think that I'm not that keen on like the discipline of ethics, which tends to be very much that there are abstract principles, it's, you know, that you can't necessarily evaluate every single condition or context based on an abstract principle. So I was trying to find like a master's program that would work for me. And for me, it ended up being this field called Science and Technology Studies. Because as I was reading the papers there, I was like, these people get it. Like they're reporting exactly the kinds of experiences I had in the lab. They are studying carefully what is going on, and trying to think about what this means for the relationship between science in society, what role the government should have in shaping science, what role members of the public should have, you know, all those kinds of questions. So that's kind of, I like sort of felt like I found my home, and then sort of have stayed in that since then. But work a lot with, so synthetic biology, they're sort of my community now. So I've like been working with them probably for 10 or 12 years, sort of as the field has grown. And so in addition to human practices, another phrase I think it's on the iGEM website quite a lot too, responsible research and innovation, or RRI. It's more of a European thing, but European funders I'm trying to think when, maybe 5 or 6 years ago, decided to make this a central element of almost all their science funding. So there's been this like massive push to incorporate responsible innovation into all of the big synthetic biology projects and things like that. So a lot of my research, some of my research is completely standalone, my own interests in synthetic biology, and then some of my research is actually working a lot with different project teams, helping you know, helping to frame projects and helping to do research that will contribute to the project in a sort of integrated human practices kind of way. So I think synthetic biology is super unusual in having this. It's not something that you tend to encounter. I don't know any other fields that have this degree of like heterogeneity within the field, that have artists and designers who are kind of big parts of the conversation, that have, you know, I mean, this afternoon I was on a panel discussing the ethics of synthetic biology at the New York Academy of Sciences. Like they constantly, there's this kind of energy and interest and motivation within the synthetic biology community to be sort of trying to figure out how to actually use this technology for good. Because it's pretty clear you need to think about that consciously these days. Otherwise, like, capitalism takes over, and you know, and there's no incentive to pay attention to, you know, environmental impact or things like that a lot of the time. So this is a community that cares a lot, I think, and is trying to figure out productive ways of doing that, which is fun for me, right? Like it's so.
People seem interested in why papers are so expensive.
I don't know, right? I mean, $3,000 is a lot, right? I sit there and it's interesting. I'm like, okay, where do I submit my journal? And do I have that kind of money sitting around to spend on an open access journal or not? ASU actually doesn't have funds for you. Some universities have funds if you want to publish in an open access journal, but ASU at the moment doesn't have any funding like that. Right, well, it's priorities, right? So total priorities. Challenges in the future for biology in the next few decades. Certainly for synthetic biology, I think the field is getting to a point where applications are starting to leave the lab and enter the real world. And we have a decent history of like industrial biotechnology products, right? Insulin is a recombinant DNA product, like a lot of medicines or some medicines. And some products are produced through industrial biotechnology. We have not very much experience at dealing with releasing genetically modified organisms into the environment. And this actually goes right back to the 1975 Asilomar meeting. They actually made a very conscious and explicit statement on the first day that their interest at the meeting was not to consider any of the social, legal, ethical implications, but just to think about the safety of the workers in the lab and the safety of the sort of, the chances of it getting out of the lab, of escaping the lab. And so the system that was set up in 1975 was entirely focused on containment. And so when you talk about the BSL, biological safety level of your lab, it's all about what containment practices do you have? What ways can you make sure the organisms stay in the lab? And so what we have never done is figure out ways for organisms to leave labs in sort of constructive ways. So I think that's a major challenge over the coming years, is like, these products are starting to become ready, like gene drives, like all kinds of interesting new probiotic drinks. like living medicines, like biosensors that actually live in the environment and can help to, you know, detect landmines or clean up oil spills and things like that in ways that, you know, we haven't really fully fleshed out and figured out yet. So I think that's that's one major challenge. And accompanying that, plastic eaters, yeah. Yeah, I mean, and lignin degrading strains. I mean, the possibilities are really like really interesting. Like there's so many cool ideas out there. There's that, and then I think there's kind of issues of we were talking about this afternoon, of public trust as well, which is like, do we trust the institutions that are making these products? Do we trust their motives? Do we trust their, you know, their intentions? Do we trust the regulatory frameworks that are in place to protect us nominally? Do we trust that if something goes wrong, it won't just fall, the risk won't all be absorbed by the end users of the product or the communities that are disproportionately affected and things like that? So I think, you know, as these technologies kind of come more and more to the point of being market ready, that whole system is going to get really tested. And that, you know, I think a lot of the time when we talk about like backlash against technologies, and GM crops is often one that we, you know, tend to focus on, it's, some of it relates to those questions of, you know, it's maybe less about whether GM crops are good for you or bad for you, and more about whether you like Monsanto or not, and more about whether you you sort of trust the systems, and whether or not you feel that they're putting small farms out of business or giving them, you know, less choice. And, you know, I mean, there's all kinds of reasons for it. And so I think those are going to be, I don't know, those are kind of big issues that each country is going to have to grapple with, I think, going forward. Oh, you want to talk about companies mining human health? Yes, absolutely. And long histories of that. I don't know, ASU has its own very troubled history with this. Are you guys taught anything about the Havasupai case? Another one worth looking up. A very famous case that was settled in court, actually, where ASU researchers took blood samples from the Havasupai and then went on to use them for projects that they had not consulted the Havasupai about. And yeah, got sued, lost a lot of trust. Yeah, so it's a case worth knowing about in terms of research ethics, particularly research ethics with Indigenous communities. So there's this real tricky bind when you're talking about international development and Indigenous communities, that sometimes researchers have the very best of intentions, but also sort of wade into very different societies that have different ways of engaging with the world. And so how to do like cutting edge genomic research with Indigenous populations in an ethical way is actually an incredibly challenging task. And these are very current issues. Like there was a framework published 2 years ago, I think it was 2018, on principles for doing ethical research with Indigenous communities. Like these are not kind of long established frameworks. These are all things that are being negotiated in real time. I've actually been looking recently. There's a company called DIYbio, if any of you are interested too, that is proposing to study genomes from isolated communities around the world, in order to uncover cool genetic traits, and claiming it's plastered all over their website that they're doing this in a very ethical way. And so it's been very interesting and challenging to sort of read through how they're approaching it and try and figure out what that means. Yeah, things would be potentially, yes. Well, actually, I mean, so my position at ASU being this kind of joint appointment between biomedical engineering and the School for the Future of Innovation Society is actually partly Michael Crow's ambitions to transform engineering education. So the thought is that they want to sort of do a bit more of this like cross disciplinary sort of weaving into engineering. The trouble is that engineering programs are so stacked full that you can't do a lot of dedicated classes on it, although BME actually does. Oh, diversity sample pools for testing medicines. Yeah, I mean, do you know women were only, it's like in the last 10 years that the NIH said that you have to include women in clinical trials for drugs. So until that point, drugs were almost exclusively tested on healthy men. And actually, even now, or now increasingly, they're tested on healthy men from marginalized communities. So the clinical trial testing centers around the US are almost exclusively located in low-income neighborhoods. Well, you have to, maybe be willing to spend a few weeks or a couple of months of your time in one of these facilities, depending on what the test, what the clinical trial is, and so if you are working a full-time job, it might be hard to do that. So they don't know if these drugs will negatively affect pregnant women is the short answer, right? So anyway, so I mean, like for me, these are like super interesting, big, really important questions that I think if you want to use technology to make a difference in the world, you sort of, it's not that you have to devote your life to them, but you have to think about them. You have to figure out sort of partners in your efforts to sort of develop your technology and think about how to make it fit in the world. And so, for me, it's like, that's the kind of stuff I like doing, is sort of figuring out how you take this idea in the lab and help connect it with this much bigger, complicated, messy system that probably isn't going to agree with everything that you're doing and might force you to question some of your assumptions, but maybe make your product better in the long run, I don't know, or not. So, but yeah, all these questions are huge.
Yeah, there were some interesting ones in there. Thank you. So I don't know if you have more you want to talk about, but it's getting to like 7, so
yeah.
But that was really interesting to learn about. I had no idea that ASU actually had that much of a history with.
Yeah. It's
yeah.
And actually, so a few spring breaks ago, I hiked down to Havasupai Falls, and down in actually in like, there's one little sort of restaurant cafe there, and on the walls of the cafe they have a whole bunch of news stories about this case. Like it's
oh gosh.
Yeah. No, I mean, it actually prompted a revision of ethics rules. So
at least something good came out of it.
yeah. And I think it was a breach of trust. So those things become really important, I think, when you're thinking about developing technologies for people and groups, is sort of making sure you have a trustworthy relationship with them. So yeah, I can send you I can send you the Rolling Stone article if you want.
Yeah,
I think it's a little hard to get a hold of. And yeah, as if you have things you want to chat about as your project ideas are progressing and things like that, just more than happy to chat. So just reach out.
I'll send the teams your emails your email if that's okay.
Yeah, that's great.
Awesome. Okay. Does anyone else have any questions for Dr. Frow or comments?
I do kind of have a question about, so I know for the COVID vaccine, they're speeding through the clinical trials to get FDA approval, because that usually takes years to get through. And I was wondering what your insight was on this, or what you thought about it. Like, do you think that will change how the FDA starts to approve things in the future? Will it not take as long? I don't know. Or do you think, of course, maybe you don't know, like, will this negatively impact the vaccine in the future? Like, will we see later there's side effects to these? I don't know.
Your questions are exactly the right questions to be asking. And like, I don't know what the answers are, right? I mean, there's a lot of public health officials right now, I think, are really worried that public uptake of the vaccine will not be that high because of a sense that it was being rushed through. You know, vaccines are a particularly thorny subject in the US, right?
Yes.
So I don't know what proportion of the population. I was talking with somebody from the World Economic Forum who said they've been doing a lot of work on this and they've been trying to figure out. So I think the Biden transition team is already trying to get like public communications experts on its COVID team to figure out how to try and change the messaging around COVID vaccines. And that someone from the World Economic Forum was saying that they've been digging into this issue to try and figure out where they could best make a difference. And that it turns out employers may be a critical kind of linchpin in all this, because people tend to, like employees tend to have on the whole decent working relationships with their employers, and that it might be that actually working closely with employers is maybe a way to promote kind of uptake and messaging around the vaccine. But like, oh, completely. And the FDA goes through these kind of big cycles too of kind of more and less regulation, right? Sort of on, they don't act by themselves, they're sort of mandated by Congress. And so sometimes they sort of enter these periods of deregulation, and then usually there's some sort of public health crisis that emerges from that, and then they go through a period of tighter regulation and so on. So, you know, I think they're great questions. It's possible that it might result in, you know, if these vaccines do turn out to be fairly effective and that speedy process zipped through, then, you know, maybe there's a phase where FDA approves things faster. Interestingly, Moderna is really a synthetic biology company. So it may turn out to be the synthetic biology success story that the community has been dying for a long time, because they don't really have a success story to point to right now. I don't know about sunscreen. It wouldn't surprise me. There are a lot of things that have been grandfathered into systems. Yeah. I mean, if every new product had to go through the same regulatory processes that they do now, we probably, I mean, I don't know if this is true or not, but people say that like aspirin would never have made it through clinical trials or something. I don't know if that's the case, but so.
Wow.
Yeah.
Thank you.
Yeah.
Yeah. Thank you.
It was great to meet you, and yeah, good luck with your projects and your brainstorming, and just fun to see that there is a student group on campus that are energized by synthetic biology.
Yes, finally.
Finally, no, I mean it's been a bit of a dry spot on campus. So all right, well, take care, and yeah, let me know if I can help in any way as your club and your iGEM projects progress. Okay.
All right. Thank you so much.
Yeah. Bye.
Thank you.
Okay.